There you go. Great, thank you. Well, okay. The second speaker is Jianfu Li from UT Houston, and he's going to talk about fine-tuned domain-specific language model and its application for vaccine ontology. Jianfu, please.
Okay. Good morning, everyone. It's a pleasure to present our work on the Cascaded framework for mapping vaccine ontology formed from clinical trials using fine-tuned domain-specific language models. The study was conducted in both Dr. Hu's group from University of Michigan and Dr. Tao's group from UT Hills. So this is the outline of the presentation. As we know, the vaccines have revolutionized the public health to provide protection against infectious diseases and cancers. To support the advancement of vaccine research, development, and implementation, the vaccine ontology has been developed as a community-based ontology to promote vaccine data standardization, integration, and computer-assisted reasoning. And notably, the clinical trials are crucial for ensuring the safety and efficacy of vaccines. It plays an essential role in their development by providing critical data. And the ClinicalTrials.gov is a valuable repository of clinical trial information. ACT is aggregated analysis of ClinicalTrials.gov is a comprehensive and publicly available database derived from the ClinicalTrials.gov registry. In this study, we use the ACT to get the clinical trials data. ACT provides detailed information on clinical trials registered in ClinicalTrials.gov. However, the vaccine information stored in the clinical trial database is not standardized with non-uniform vaccine names, abbreviations, and codes, which may lead to confusion and errors in evidence-based decision-making and vaccine surveillance. So normalization is a very important step for vaccine names to improve the consistency and clarity of vaccine group names. Manually harmonizing the full clinical trial information into the clinical trial database is time-consuming and resource-intensive. There's a very few studies on the normalization of vaccine names, and until now, there's no research conducted on standardizing the names of vaccines from clinical trials to align with the vaccine ontology. Therefore, there's an urgent need to standardize it and link the vaccine name method in the clinical trial to standardize the ontology. So here are the three goals of this study. First, we try to fine-tune multiple domain-specific language models to map vaccine ontology from clinical trial. Second, we try to propose a weighted rule-based ensembles to enhance the mapping performance. Third, this study supplements the concept of coverage in current VO to enrich its vocabulary and further enhance its interoperability with other ontology. Here's the Cascaded framework snapshot in this study. The more details will be described in the following slides. So for the data set, we mainly utilize the three publicly accessible data sets from the website. First is the VO ontology, the vaccine ontology. We downloaded the vaccine ontology data from the official website. For the ACT database, we also downloaded the database directly from their official website. And for the ACT database downloaded, since it contains the original clinical trial information, which contains the main column like the interventions, we mainly extract the vaccine names from the intervention column in the ACD database. The third dataset is the UMLS 2023 full release. Yeah, the Unified Medical Language System is a set of files and software that brings together many health and biomedical vocabularies and substandards to enable the interoperability between computer systems. We mainly included this dataset for further fine-tuning our large language models. So for the vaccine annotation, 2 domain experts helped to do the annotation, follow annotation guidelines to annotate from clinical trial to vaccine ontology. Firstly, a joint annotation for 150 vaccine terms from CT by the 2 experts to help to get the agreement. And then each expert was assigned 200 vaccine terms to annotate independently. So finally, we get 550 annotated vaccine corpus for our further model training and evaluation. In this study, for the fine-tuning, we initially pre-trained the PubMedBERT. You can see here in the second figure, the CTPubMedBERT. On the clinical trials, and are typically used in medical concept normalization. By leveraging the developed corpus, like the clinical trials, we aimed to enhance the model's understanding of vaccine-related language and concept. To further improve the model's performance, we moved on to retraining the SAPBERT. SAPBERT is a pre-trained language model based on PubMedBERT that focuses on self-alignment to learn representations of biomedical entities from UMLS corpus. It achieved new state-of-the-art results across 6 widely used benchmark datasets for biomedical entity linking. So here we retrained our model based on the SAPBERT and the latest UMLS 2023 release data. Then finally, we further fine-tuning our final model using the vaccine ontology knowledge and clinical trials vaccine information. This is resulted in our CTPubMedBERT plus SAPBERT plus VO model in the last column of the figure, the fine-tuning. To further enhance the performance, we propose a weighted rule-based ensemble method. The ensemble method assigned different weights to the top 3 models prediction. So it's given more importance to the models that demonstrate a better performance. By aggregating the knowledge and the insight from these models, we aimed to achieve higher accuracy and robustness in the normalization of vaccine-related data. So here is the evaluation formula in this study. We utilize the top N accuracy. It quantifies the proportion of currently predicted concept relative to the total number of concepts predicted by the system. Here's the results of the data processing and screening. We utilize the PRISM procedure. This figure shows the results. We can see from the top of the figure is utilize the 2 ways to get the vaccine names from the original clinical trial. In the left side, we try to search in the ACT database using keywords directly. So it gets 6,472 records. And on the top right, we try in the other direction. We first get the vaccine names in the vaccine ontology. Then using those vaccine ontology to search in the ACT, then we get the 3,012 records. Finally, we combine them and remove the duplicate records. And then we get 7,873 records in this study. Here is the result for the top-end accuracy of mapping VO from clinical trials using our proposed approach and the baseline, the BERT-related models. We can see from the top table, it's a fine-tuned domain-specific models. There are 8 fine-tuned models, like the baseline BERT, the PubMedBERT, and the third is the SAPBERT plus PubMedBERT. This is our retraining the SAPBERT based on the latest UMLS dataset on the PubMedBERT. And then the CTPubMedBERT plus SAPBERT, and the SAPBERT plus PubMedBERT+VO. This is our fine-tuning based on the SAPBERT and the PubMedBERT using the vaccine ontology. Then is the CTPubMedBERT plus SAPBERT+VO. And this OMAPNet is another publicly trained language models, which achieved the best performance in a benchmark compared to other models on different natural language processing tasks, such as the normalization. We also included those other baseline. And the last fine-tuned model, or MapNet based V2 SAP CTVO, this is our fine-tuned using the vaccine ontology knowledge and the clinical trials based on the MapNet model. So the bottom table is a weighted rule based on ensemble result. It resulted in 6 different different performance. Finally, we can see in the last 9, the ensemble plus scale plus stream matched rule achieved the best performance. And compared to the fine-tuned model, we can see that our ensemble method significantly improved the performance of the normalization from CT to VO. So for the error analysis, we first classified major error types like the NER, the name entity recognition, abbreviation, disambiguation, hierarchy, semantic stemming, etc. There's different major errors. And the error analysis was conducted on the top-ranked predicted concept generated by the proposed approach. The figure shows the different percentage of the errors among the four tested sets. So the contributions in for this study lies in 3 aspects. Firstly, we propose a cascaded framework to utilize fine-tuned domain-specific language models. Second, the cascaded framework can be seamlessly integrated into existing ontology platforms. It enhances the performance of concept mapping. It also provides an advanced approach to medical concept normalization. Third, this study addresses the issue of ununified granularity in vaccine ontology terms. It enriches and refines the concept within the ontology. So the impact of the fine-tuned domain-specific language models is showed that a significant improvement in the accuracy of the mapping process was achieved by utilizing the multiple domain-specific knowledge. And the impact of the weighted rule-based ensemble. The progress achieved through the implementation of the string matching rule in the ensemble method effectively addressed the primary obstacles encountered in the VO normalization task, such as the variability and noise found in vaccine names extracted from the interventions in the clinical trials. So the limitation in the study, firstly, our study currently relies solely on data from clinical trials. We may expand our data to different domains. And the availability of a larger amount of annotated data is also limited, since the annotation takes time. So yeah, in the future, we may try to annotate more data to improve our model. For the future work, we are going to improve the variability of the vaccine names, both in vaccine ontology vocabulary and clinical trials. We also will introduce a lambda entity recognition step to remove noise from the original data in clinical trials. Since in the original clinical trials, the intervention columns may contain many noises and non-standard terms. So a fine-tuned NER model may help to improve the extraction to get more exact vaccine data for our mapping. So these are key acknowledgements. Thanks for the project members from Dr. Hu's group and Dr. Tao's group, and Dr. Hu, Dr. Zheng, and Yuan Yi, Pan, and Jin Jingguo. Also Dr. Tao's group, Dr. Tao, Dr. Fang Li, and Yiming and Zhou Lan. Also, thanks for the project funding from NIH grants. Thank you.
Great, thank you. Okay, well, do we have any questions from the audience for Dr. Li? Quiet. Dr. Li, just quick question. Have you considered or have you tried using a larger language model on this specific test?
In fact, we tried, but the performance is not good for the normalization. Since, for example, we tried to using the ChatGPT to do the normalization from CT to VO. We prompted the ChatGPT with like something, find the vaccine names in this clinical trial text. Then and also we try to ask the ChatGPT to output the result in a structured format. It can do that, but the result not good. Is inconsistency. For example, it can show some vaccine names and the link, but those are not the real link and the real vaccine name, something like that. So we finally didn't include in the large language model the performance and experiment in this study. But yeah, we may try to do the large language model in the further future study.
I see. Yeah, there are issues with inconsistency and how you structure your prompt. I mean, you get very kind of different results. And we actually use ChatGPT. I mean, Hashin's going to present a little bit about it later today. So maybe we can discuss further later.
Oh, that's great. Yeah.
Okay. All right. Do we have any other questions? If not, I think we can move on to the next one. Thank you, Dr. Lee.
Thank you.